Hello, welcome. In this video, we are going to go over cell reproduction, or simply put, mitosis and everything that goes into mitosis. So let's go ahead and get started. These are our objectives for section 10.1, cell division. We're going to describe the structure of prokaryotic and eukaryotic genomes. We'll talk about what chromosomes, genes, and traits are and how they are different and how they're related. And then we will talk about how chromosomes compact because they're so big and how do they fit inside of a cell. So just some things to think about before we get into cell division. True or false? A cell in your liver, brain, bicep, muscle, and skin on your foot all have the same DNA. Now you might think, well, that should be false, right? Because liver, brain, bicep, muscle, skin, they all do different things. It's actually true. So your DNA is the same throughout your body, at least in your body cells. Now, of course, there is an exception for your gamete cells, like egg and sperm. But if it's not an egg or sperm cell, it's going to have a certain set of DNA, and that DNA is going to be the same in every cell in your body. It's just which genes are activated, which traits are activated in that DNA in that specific region, which makes that cell do different things. Let's think about this starfish. So the starfish lost its arm, and they are able to regenerate its arm. So by what process are the missing cells replaced? Now you might think, well, we haven't talked about starfish and how they regenerate their arms. Well, what simple process are they going to do? They're going to do cell division to replace that arm. So they're going to have to make more cells to get to that arm shape that they had before. So they're going to do cell division, which results in more cells. Same thing happens when you get a scar or when you get a wound and your body starts to replace the missing skin and you end up with a scar. Okay, so cell division has two functions. Multicellular organisms use cell division for growth, for maintenance, and for repair. So we just talked about how multicellular starfish can repair their arm by cell division. They can also grow, right, and then maintain. So if there's a cell that's maybe acting finicky, they're going to get rid of it and then just replace it with cell division. The body's going to get rid of it. For single cellular organisms, they're going to use cell division to reproduce. So if we think about just one cell, maybe like a bacteria cell, they're going to divide and that ends up in two organisms. For egg and sperm cells, they are produced by single cell division as well. Okay, the cell's DNA makes up its genome. So whichever ATCG bases are recorded in whichever order, they're going to make up traits, and those traits are the genome. And in single cellular organisms, their genome is a single chromosome. It is circular, and it's residing in the nucleoid region. So this is just an area inside of the cell, inside the single cellular organism, where you could find the DNA. Additional smaller loops of DNA are called plasmids. So you might have seen this here. Plasmids are picked up from other bacteria cells, or they can be picked up from the environment, and they usually have a few genes on them. A lot of the genes that they have allow them to be resistant to like certain antibiotics or certain like antibacterial soap. So plasmids are very beneficial for the bacteria, not beneficial for us if we want to get rid of them. All right, so the eukaryotic genome consists of several double-stranded DNA molecules, which are chromosomes. So can you think about what a chromosome looks like? So let's take a look at this, this plant root over here. So each of these really thick structures here, these are each chromosomes lined up. They're going through cell division. And so we can kind of get an idea of what a chromosome looks like. So a bacteria chromosome is circular, but a eukaryote chromosome is linear. So make sure you make that note in your notes. So a prokaryote has circular DNA and a eukaryote has linear DNA. And we, eukaryotes like us, we have multiple pieces of DNA that makes up our genome. But we'll get to that in a second. Okay, so you heard me talk about like your liver cell, brain cell, bicep muscle, skin on your foot. Those cells are all going to have the same DNA, right? And they're going to have two sets of their DNA. So they're called diploid cells. It means that there are two. So if we think about like a skin cell or a liver cell, two sets, right? So there's two of those, 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 two of everything. In a gamete cell, so egg or sperm cell, you're going to have half the number of chromosomes. So notice there's only one copy of each chromosome. And this is totally random on which one gets picked. So if we take a look at these two right here, notice there's a little yellow mark on that one, there's a red mark on that one. So the one that got chosen for this gamete cell is yellow. If we take a look at this one, yellow and red, well, the red one got chosen in that case. So it's totally random on like which of the two in the set get chosen for that gamete cell. And that's totally okay because we want some differentiation in our gametes, right? You don't want your brothers and sisters to look identical to you. And so they're going to have different chosen pieces of their DNA. So that's how you look different from your siblings. It also, not only does it result in different appearances, but it results in different other traits that we don't really see. So could be like you're disease prone or not disease prone, or you have maybe a sickle cell versus no sickle cell. So things that you can't see but are definitely apparent. Okay, so eukaryotic chromosomes, here's a better look at what they look like. This is a snapshot of the human genome. So in every cell in your body that is a somatic cell, right? Somatic cell, in every somatic cell, this is in the nucleus. All of these chromosomes are in every cell, every somatic cell in your body. So you have 23 pairs of chromosomes in a somatic cell, which sounds crazy that you have that much DNA. So we're going to get into how we compact that DNA here in a second. But I do want to point out that humans have 23 pairs. That's not going to be the case for every organism. It differs for each organism. I don't have a specific example, I wish I did, but it's not going to be 23 for every organism. I know a fruit fly it has like 7 pairs of chromosomes, so not quite as many. Okay, poll question. A diploid cell has blank the number of chromosomes as a haploid cell. So let's think about our vocabulary. A diploid cell has, what, twice, letter C, the number of chromosomes as a haploid cell. So diploid, we can write it as 2N, and then haploid is just N. All right, so more about chromosomes. Chromosomes that pair up are called homologous chromosomes. So in our human genome here, these chromosomes on number 1 are homologous. They pair up. The chromosomes here for number 2 are homologous. They pair up. The chromosomes for number 3, they're homologous. They're a homologous pair. Okay? So they typically have the same length, or roughly the same length. They typically have the same centromere location, so the center. I know these probably look a little different than what we've been looking at. Same length, same genes, but not necessarily the same allele, same gene loci. So on one pair of chromosomes, let's say this is the gene location for blood type, okay? So you're gonna be A, let's say we're A, and then this one is B. So they're not necessarily gonna be the same allele, meaning that that's the same variation of that trait for blood type, but the blood type trait is going to be on the same location for the homologous pair of chromosomes. Okay, so we're also going to see chromosomes that look like this. Now I know you might think, well, there's like 4 here. Well, technically there's only 2. So chromosomes will look like this after a certain stage in interphase in the cell cycle, where they double their DNA, so they make a copy, and that copy kind of sticks with them, and it ends up looking like an X. So this half of this chromosome is identical to this half of this chromosome. So that ensures that everything's copied correctly and the resulting cell gets the correct DNA, right? All right, so you're going to see chromosomes that look like this. Again, this is just one chromosome here, even though it looks a little different. When they look like this, we call them sister chromatids. Okay, so this is a chromatid here, and then these are sister chromatids. Okay, so just kind of make that note. I know there's a lot of vocabulary for this chapter, so just try to keep up with that. All right, so some genomes have pairs that don't match. For example, the X and Y chromosome here. So X and Y is number 23. That determines our sex, so male, female. Females are X, males are Y. If there's a Y present, it's automatically a male. If there is no Y present, automatically a female. And I say it in that term because you can have just one X, and that would still make you a female. Or you could have 2 X's and a Y, and because you have a Y, it makes you a male. So those are just some weird variations that can happen, but we're going to talk more about genetic differences in the coming chapters. All right, so how are chromosomes compacted into the nucleus? Well, it actually is very simple. This is our regular DNA helix, right? So on every chromosome, this is what it looks like visually. And these pieces here in the center, blue, yellow, green, red, those are the bases, A, T, C, and G. Or they're representing the bases, right? So this is our DNA helix here. And then in the center, looks kind of familiar, right? Looks like a protein. So this is called a histone. It is a protein, and it helps DNA compact itself, and it keeps it organized. So if you think about like your headphones, the wired kind of headphones, and you put them in your pocket, they're just going to get really tangled up in there. Well, putting them on, wrapping them around something will keep them organized and they won't get tangled up, correct? So same thing goes with DNA. So if we just wrap them around this protein, histone, then we can keep it organized and it's not going to get all tangled up. So that's going to happen a lot of times over and over. So coiled up onto these histones, and then the histones compact on each other and they coil, and then they further condense into a chromosome shape like this. So this is super, super, super dense. And this is how a chromosome will look at certain points of the cell cycle. The DNA is not always going to be so compact, but it helps that it's compact during cell division so that we ensure the cells get the correct DNA. Okay, so during other points of the cell cycle, like this cell here, its DNA is not so compact, and that helps it keep it readily available for the cell to use, right? Because the cell has to read the genes that are encoded on DNA. So if they're compacted all the time, they're going to be really hard to read. So after cell division happens, the chromosomes relax a bit, and that allows them to be accessed correctly. Okay. All right, so there's a really cool video here if you want to watch on your time, and there's a nice meme that I like to show my students. But let's keep moving. Okay, so that was 10.1 cell division. Hopefully now you can compare, contrast prokaryote, eukaryote genomes. Hopefully you can tell me what a chromosome, gene, and trait is. So chromosome, again, is that big structure that we were just talking about, the compacted one. Genes are things like blood type that we talked about. And then the trait is pretty much a gene, but the trait could be referring to like your blood type A or your blood type B, so the variations. And then now hopefully you can describe the mechanisms of chromosome compaction. Okay, so 10.2, the cell cycle. Describe the 3 stages of interphase. We're going to talk about chromosomes during mitosis. So we'll talk about the phases of mitosis, and we'll talk about how cytoplasm is divided and what the G0 phase is in the cell cycle. There's a nice little website here. It allows you to access this interactive for 5 minutes for free. And that goes over cell division. If you just search Gizmo cell division on Google, that'll just show up there. Okay, so the cell cycle looks like this. And notice that mitosis is a very, very small part of the cell cycle. So most of the time when you see a cell, or you think of a cell, it's going to be in interphase. So this entire portion here that's not peach is interphase. And the interphase is divided into 3 sections. One is G1, then there's an S phase, and then there's a G2 phase. So again, G1, S, G2, it's all interphase. Okay, so let's go ahead and talk about each of these phases here a little bit more. This is a really cool animation of cells dividing. It just shows you how fast that they do it. And notice the cells around them, what are they doing? They're resting in interphase. Okay, I could watch that all day. Alright, so the cell has 2 major phases. So interphase, the gray phase here, and then the mitotic phase, this peach phase. So the mitotic phase is where those steps that we were just watching in that video happen. And again, it happens so quickly, and then it's over, and then the cell enters interphase, and it does these things here. So let's talk about what happens in each phase of interphase, or each stage, I guess, of interphase. So the first one is G1. So let's say the cell that we're talking about, it just divided, it just came out of mitosis, cytokinesis, and now we're in G1. So the cell is going to be kind of small because it just came from division. So all it's going to do is grow. We call it gap 1 phase. So we call it gap 1 phase because scientists thought that nothing really happened during this phase. So we just call it gap phase. It's gap 1. But actually the cell is growing, it's taking on nutrients, and it's trying to get back to a regular size. The next phase is called S phase, which is short for synthesis. And this is when we are going to copy the DNA. So we're going to go from a single chromatid to sister chromatids here in this phase. So just like this. So you have a chromatid, and then there's a chromosome. So we're going to have a chromatid, we're going to make a copy of that chromatid, put it together, it's a chromosome. In G2 phase, what's going to happen is the cell is going to continue to grow, and it's going to try to duplicate the organelles inside of the cell just so that the resulting two cells, after the mitotic phase, the resulting two cells will have enough organelles to continue on with their cell cycle. Okay, so let's put some things together here. So matching, chromosomes are duplicated. Which stage is that? Well, that should be S phase, right? So B. Accumulating biomolecules should be A, G1. And then final preparations before division, G2. Good job if you got those right. Let's answer this question down here. Which of the following events does not occur during interphase? What do you think? Should be letter D, right? So separation of sister chromatids occurs in the mitotic phase. So DNA duplication occurs in S phase of interphase. Organelle duplication occurs in G2 of interphase. And increase in cell size occurs in G1 of interphase. All right, the mitotic phase involves several steps. So we're going to go over each of the mitotic steps in the next several slides. So mitosis is sometimes referred to as karyokinesis or nuclear division. And this is what mitosis is. We're really just dividing the nucleus. Cytokinesis is when the rest of the cell divides. So when the cytoplasm divides and the organelles inside are divvied up into the two new daughter cells. That's cytokinesis. All right, so we're going to talk about the different stages of mitosis. Maybe you can pause it here and name these stages before we even go over them. If you can, good job. If you can't, come back to this and try it when we're through talking about the stages. All right, so here are the stages: prophase, metaphase, anaphase, telophase, cytokinesis. I did skip prometaphase. We'll talk about what happens during prometaphase. I don't really talk about it much because I usually group prophase and prometaphase together. All right, so prophase is the first phase that happens during mitosis. So we have just come out of G2, right? So we were in interphase. We went all the way through. We ended at G2 with interphase, and now we're in the mitotic phase. So this is prophase. The nuclear envelope will start to break down because we have to access the chromosomes so that we can divide them correctly. The things called centromeres, remember from the chapter we talked about cells. So centromeres, centrosomes will begin to divide and migrate to the poles. And they start to create spindles. So the spindles are going to help connect, or they will connect to the chromosomes, and they will help divide the chromosomes separately into resulting cells. So another part of prophase is prometaphase, and really all that happens is that the spindles start to connect to the centromere region of the chromosome. So connecting the spindles to the centromere will help divide them. So they're going to split here in a second. All right, in metaphase, we have a check during metaphase. So this is called the M checkpoint. We're going to check to make sure that all of the chromosomes are connected to a spindle. So all the centromeres are connected to a spindle on each side. So remember, during S phase, we had a chromatid, and we copied that chromatid, and now we have sister chromatids. So let's say this is chromosome 1. This is one copy of chromosome 1. This is our other copy of chromosome 1. So now we're going to line everything up in the center: chromosome 1, chromosome 1, chromosome 2, chromosome 2, and so on. And then once we start to split them, now each resulting daughter cell is going to have a chromosome 1, chromosome 1, chromosome 2, chromosome 2, right? And then same thing for this side. So lining them up in the center and making sure that all the spindles are connected correctly will make sure that the two resulting cells are identical. So remember, let's talk about it again. This chromosome copied itself to make this one during S phase. And if we split them apart, the genes that are going to this side are the same as the genes that are going to this side. Okay. So this phase is called anaphase. So this is when the chromosomes, sister chromosomes or homologous chromosomes split, or sorry, sister chromosomes split. And then telophase is when the chromosomes have divided themselves properly and the nuclear envelope starts to reform. And this is when cytokinesis occurs. And cytokinesis happens a little weird, a little bit different in different cells. Cytokinesis in animal cells, there's a ring of microfilaments that produce or create a cleavage furrow here, and they pinch the cell off. And then in plant cells, they don't have the microtubules like animal cells have, and so what they end up doing is creating a cell plate, and that divides the cytoplasm in half. And I know this is probably confusing. I don't know why they made them X-shaped here in the image, but just think of them as microtubules there. All right, so can we put these in order, and can we name the phases? So go ahead and pause it. We're going to go over the answers here. So the first phase, or number 1, should be this one right here. So this is going to be what? This is going to be prophase. So it's not interphase because the cells, the chromosomes are condensed and they are, or the nuclear envelope is starting to break down. The centrioles are starting to produce their components, their spindles. So this is going to be number 1, prophase. The next one is this guy. So this is number 2, and this is metaphase. So you think metaphase middle. Number 3 is this one. So this is anaphase, as the chromosomes are separating, the chromatids are separating. Number 4 is this phase. This is telophase. The nuclear envelope is starting to reform. And then this phase here is cytokinesis. This is number 5. Cytokinesis occurred and the cytoplasm has split. And you can notice that the chromosomes are starting to unfurrow and relax a bit. So great job if you got those. If you didn't, it's okay. There's a lot more practice here in a second. Let's talk about what G0 phase is really quick. So G0 phase is part of the cell cycle, and we haven't seen it on the other images because it's not that it's rare, but there's only a few cells that really stay in G0 phase. Usually the cells that stay in G0 phase and don't come out are like your mature cardiac cells, so your heart cells, and nerve cells. So they enter this phase of quiescence, or phase of inactivity, and they stay there. So they're not going to duplicate their DNA, and they're not going to divide anymore. They're just going to stay here. Cells can enter G0 and leave if they get the necessary requirements back. So they can enter G0 due to like environmental conditions, nutrients availability or unavailability, and growth factors. So if they get those things back, if environmental conditions improve, then the cell will exit G0 and enter G1 again. Alright, so let's identify some of the stages of mitosis here. So let's see, we'll start with the top here. So the yellow one is anaphase. The black one is metaphase. The red one is prophase, or sorry, I want to say the red one's probably interphase. The green one is probably interphase as well, and the blue one looks to be like metaphase because they look like they're lined up in the center. Alright, this one image is a little bit more clear. The yellow one is telophase, or maybe late anaphase. Green is prophase, so the chromosomes are condensed, we can make them out, but they're not really like lined up yet in the center. So prophase. This one is going to be interphase. This one is metaphase. They're lined up in the center. And then this one is anaphase. So great job if you got them. There is a video here if you want to watch. It's a short video on how mitosis occurs and what the cell stages look like. And there is a quizzes that goes over the phases of mitosis that you can practice with. Okay, so that's 10.2, the cell cycle. Now you should be able to talk about G1, G2, and the S phase, or if I say them in order, G1, S, and G2 phase of interphase. Hopefully now you can discuss the behavior of chromosomes during mitosis, and you can explain how the cytoplasm is divided, especially in plant and animal cells. And then hopefully you can talk about the G0 quiescent phase. And again, there's that gizmo that goes over each of those phases. Okay, 10.3, we'll talk about the control of the cell cycle. So hopefully you'll be able to understand how the cell is controlled both internal and externally. Hopefully you can explain how the 3 control checkpoints occur. So we'll talk about that checkpoint. I know I talked about the M checkpoint already. And then we'll describe molecules that control the cell cycle. Alright, so control the cell cycle. External triggers can initiate or inhibit the cell cycle. So this can cause the cell to go through apoptosis, which is controlled cell death. They can release growth hormones, which makes the cell grow and go through mitosis. And then also cell crowding can inhibit mitosis, so that could cause cells to enter quiescent phase if there's too many cells. So if you have a wound, if you have a cut on your skin, and the cells are not touching each other there in your skin, they will be triggered to go through mitosis. So they'll exit the quiescent phase and then go through mitosis and keep going through the cell cycle in mitosis until they are crowded again. So as cells begin to touch each other again and get crowded, they will slow their cell cycle down and maybe even enter quiescent phase. Alright, so let's talk about different checkpoints inside of the cell. So these were all external triggers. So now we're going to talk about internal triggers. So there are checkpoints. There's a G1 checkpoint, a G2 checkpoint, and then a mitosis or an M checkpoint. Okay, so we're going to talk about each of these on the next few slides. The G1 checkpoint determines if the conditions are favorable for cell division. So if the conditions are not favorable, what's going to happen? We just talked about it. It's going to enter G0 phase, right? So if there's bad conditions, we're not getting the nutrients that we need, we'll enter G0 phase. Okay, so if G0 phase, or if the G1 checkpoint is good and the cell meets all the requirements, it'll move on to the S phase where the DNA is synthesized. and then it goes all the way through S phase and enters the G2 phase before the next checkpoint. So the G2 checkpoint helps to ensure that all of the DNA has been synthesized, and if it hasn't been synthesized, it'll stay here until it has been synthesized. And if there are any errors in DNA replication, they will be repaired. If there is any damage, those damages will also be repaired before it enters the mitotic phase. So you might be thinking, well, this is kind of like silly. Won't the cell, you know, won't the cell know that it's done something wrong? Well, this is how it knows it's done something wrong, is by these checkpoints. You don't want a bad cell going through the mitotic phase, because then you'll have more and more bad cells, and that's how cancer happens. So we'll talk about cancer in a second. All right, so the last checkpoint, M checkpoint, makes sure that all of the tubules are connected during mitosis during metaphase of mitosis. If they aren't, what happens is non-disjunction. So one cell is going to have extra DNA, and the other cell is going to be missing some DNA. All right, so match these checkpoints to their descriptions. All right. So number 1 should be C, energy reserves for S phase. Number 2 should be D, assessment of new DNA. And then number 3 should be B, attachment of spindle to kinetochore, or to the centromere or centrosome. So letter A and letter E were not used. So let's talk about those. So letter E, synthesis of DNA, that does not have a checkpoint. That's just S phase of interphase. Letter A is going to be anaphase of mitosis. So separation or segregation of sister chromatids. So there's not an S checkpoint because there's a checkpoint at G2 that checks the DNA for the cell. So it would be silly to have an S checkpoint and then a G2 checkpoint that do the same things. Okay, so some other things that can be regulators of cell division are positive and negative regulators. So these are things internal to the cell and also external sometimes. So positive regulators promote the cell to move to the next step. Negative regulators stop the cell. So when we reach a checkpoint, like G1 checkpoint, if the cell is good and it has accumulated the correct nutrients and materials, then the positive regulator is going to promote the cell to the next phase of interphase. If, however, there's something wrong, there's bad environmental conditions, then a negative regulator will act and it will stop the cell from advancing. So these are just some examples, specific examples of positive regulators. So cyclins or CDKs are some of the positive regulators in cells. And there are certain ones that are active during certain stages of the cell cycle. And so this is how we can track, or this is how we figured out that there are checkpoints during the cell cycle. You don't have to memorize these, but I thought it was just interesting to see that there's specific molecules and we know when they're active. Negative regulators, so these are also some specific examples. RB, P53, and P21, they primarily act at the G1 checkpoint, so they're going to stop the cell and maybe force it into G0 phase or just halt it at G1, and it won't go on to the next phase. Okay, so that's 10.3, so that's how the cell can regulate itself during the cell cycle, just so that it doesn't go on and have uncontrolled growth. So let's talk about what happens when there is uncontrolled growth. That's cancer. We'll talk about oncogenes and proto-oncogenes and tumor suppressor genes and what they are and why they're involved in cancer and how. So cancer and the cell cycle. So the term cancer is very broad. It covers a lot of diseases characterized just by uncontrolled cell growth. So a benign tumor can come about by uncontrolled cell growth. So you can somewhat call that cancer. Or you can have malignant melanoma, which is really bad uncontrolled cell growth, and that's also cancer. So cancer is just kind of a big umbrella term. Cancer begins with a gene mutation. So something happens in the DNA where it's mutated, and that results in faulty regulation of the cell cycle. So we just talked about positive and negative cell regulators. Well, a mutation occurs in the genes that code for those regulators. So if there's faulty regulators, then there's going to be a faulty cell, and then if there's a faulty cell, that cell's going to go on and make more faulty cells from itself. And that's when we get a tumor or a lump. All right, so proto-oncogenes are normal genes that code for positive cell regulators. So proto-oncogenes are what's in your DNA, and those proto-oncogenes are coded, and they're made into proteins, and then those proteins act as positive cell regulators. So remember, proteins do a lot of things. Proteins are used throughout your body for a lot of different jobs. They do the hard work. This proto-oncogene, the segment of DNA, codes for just one protein, and this protein is a positive cell regulator. So normally, this positive cell regulator, when there's damage or abnormalities in the cell, then the cell will be arrested. I like that term, arrested. The DNA will be repaired, or it will be programmed to die. And if there's a mutated version of this positive cell regulator, then if there's abnormalities in the cell, it's not going to catch it. And so it won't arrest it, and it won't stop the cell from dividing. So we're going to continue to get bad cells over and over. So if we let one cell through, then that one cell turns into 2 cells, and then those 2 cells turn into 4 cells, and then that's how we end up getting a tumor. Tumor suppressor genes are other genes that suppress tumors. They do what they sound like. So this is the gene that is encoded in your DNA, and then they code for negative regulator proteins. So normally, a negative regulator is going to stop the cell from dividing or from continuing on if it's faulty. But if there's something wrong in the gene for the tumor suppressor, for the negative regulator, if there's something wrong in that gene, then that negative regulator is not going to be made correctly, and it won't function correctly, and the cell will just continue on with the cell cycle even though it's faulty. Okay, so these are just some things to think about. I'm not going to go over all of these answers, but what is a negative cell regulator, like P53? P53 halts the cell if it's faulty, right? Okay. All right, 10.4, that's it. And we're going to move on to the last section, 10.5, which is just prokaryotic cell division. So prokaryotes don't go through all the phases of mitosis like eukaryotes do because they only have one chromosome, okay? So because eukaryotes have all those linear chromosomes, we have to go through all these steps to ensure that the chromosomes are divided equally in the 2 daughter cells. In prokaryotes, there's only one chromosome, so once that one chromosome is copied, then we can just divide the cytoplasm up and make sure that the 2 copies of that chromosome are in the resulting cells. And those cells are going to be identical, just like the eukaryotic cells. However, unlike eukaryotic cells, the two resulting cells are going to be separate organisms now. Okay, so that's just something different with prokaryotic and eukaryotic division. So these are the actual steps. I'm not going to focus on what happens in each step, but I do want to point out these three things. So there's an origin point, or the start of replication, and notice that replication goes around on each side. So from this origin point, the DNA is replicated both directions from that point. So this creates a second loop of DNA, and then those loops, the origin point, they will migrate towards the poles of the cell, and then those poles, once they reach the poles, the cell divides, and these things called FTSZ proteins pinch the cell membrane, kind of like our animal cell did. It just pinches it off. Alright, so there's a nice video that goes over binary fission there, and that is it.
So let's start by talking about...
Whoops, sorry, that is it. So that's all I have for chapter 10. Hopefully now you can describe what prokaryotic cell division is, what eukaryotic cell division is, and then what cancer is and why cancer happens. Alright, see you later.